Thank you very much for your presentation, Manuela. I would like to thank also to the organizers of this symposium for inviting me to give a talk. It's always a pleasure to come to France, and particularly to the Val de Loire, to remind old times with friends. Yes, the title of the talk is Chaperoning Essential Proteins, and these are the key points that I'm going to talk about very briefly, of course, during this presentation. So that hormones are messengers that transmit signals from during the process of life development and so forth, and they have to travel long or short, very short distances to get the target cell. They are very specific. For instance, hormone A, which was secreted far away, for instance, in the pituitary gland, in the hypothalamus, they travel through the body and find the target cell, which is very specific for hormone A, and here is for hormone B, and there are some cells that may respond to the same hormone or to both hormones. And this is just a figure to show that in endocrinology we have many complex systems. And as a reproductive endocrinologist, I have been interested in, of course, in studying how is the control of the gonadal function in women and men, and the pituitary, the hypothalamic pituitary axis is composed of several organs, including the hypothalamus. The hypothalamus synthesizes and secretes a peptide, a decapeptide, 10 amino acids called gonadotropin-releasing hormone. And the gonadotropin-releasing hormone, what does is travel to the pituitary and then stimulate the secretion of two hormones that are very important. They are critical for the gonadal function, which are the luteinizing hormone and the follicle-stimulating hormone. And they travel through the bloodstream, in the bloodstream to get the ovary, and then in the ovary, these hormones induce the follicular maturation, the ovulation, and so forth. Whereas in the testes, they provoke the sex steroid production, testosterone, spermatogenesis, and so forth. So these hormones are essential for reproduction. Without these hormones, any of these hormones, it is impossible to reproduce. This is a beautiful picture which was published in Science some years ago, in which this is a cryo microscopy, a cryo-electron tomography, in which we can see different organelles in the cell. This is the endoplasmic reticulum. In the endoplasmic reticulum, the proteins are synthesized. And this is the Golgi apparatus. In the Golgi apparatus, which is divided in three sections, the hormones, the proteins, are processed. The hormones, proteins are added, carbohydrates, for instance, and other post-translational modifications that are necessary for their action. And then these mature proteins may go to the membrane, the cell surface membrane. They may go to other side in the cells, or they may be secreted to act in interior cells or to interact with enzymes or whatever. I mean, the problem is that sometimes these proteins are not well conformed. I mean, they don't reach a three-dimensional structure which is necessary for be active. And this is just to show very briefly what happened. This is the endoplasmic reticulum with the ribosomes, which is this area, Okay? And they synthesize the proteins, and the proteins need to be folded to acquire three-dimensional configuration. If they are misfolded, then they aggregate, and they may accumulate and eliminate it, or degenerate in amyloid structures, which are very toxic. But chaperones, what chaperones do is to help to get the correct conformation to the protein. This is the conformation, the correct conformation, and this is a protein misfolded and aggregated. So that these chaperones are actually part of the quality control systems, and they are key factors involved in the quality control of the cell. And the first chaperone was synthesized, not synthesized, it was discovered in the 80s by the great and unfortunately late Sue Lindquist, who described the features of heat shock proteins as chaperones to help the cells, to help the proteins to fold properly. So if these misfolded proteins accumulate, they may provoke amyloidosis, formation of amyloid, and what we call conformational diseases, which include Alzheimer's disease, Parkinson's disease, and type 2 diabetes mellitus. So that if all proteins in some degree are misfolded, and these misfolded proteins usually are eliminated in a system called proteasomes. But other proteins accumulate and they provoke disease. Now let me concentrate in the GPCR. These are G protein coupled receptors. They are very abundant in the body, and they are very important because they are involved in many functions. There are almost 1,000 receptors coupled to G proteins. And this is the a general structure of the receptor and of the G protein coupled receptors, and they have all the receptors that conform what we call the family A, have very common structures that are present in almost all the receptors belonging to this subfamily. But actually, the conformation of the receptor is like this. This is a counterclockwise orientation of the receptors, and they are tightly bound to what we call the G proteins. These G proteins are activated when the ligand, when the agonist binds the receptor, then these G proteins are signal transducers that transmit the signals to the inside of the cell. So these proteins, these receptors are extremely important in our system, and in fact, 4 to 5% of the human genome codes for these kind of receptors and have a large variety, structurally speaking, of ligands, including light, odorants, peptides, lipids, proteins, etc., etc. Now, the importance is that currently, G protein coupled receptors, I will call them GPCRs, are the target. 60 or 70% of these receptors are targeted by approved drugs. So that, and they are involved in the treatment of many diseases, including asthma, cancer, heart failure, prostate cancer, and fibrosis, and an array of endocrine diseases. And here, just to show that the GPCRs transmit signals to the interior of the cell and activate the G proteins, which in turn trigger signaling cascades. Okay, so that the cells have quality control systems that in which the molecular chaperones play a critical role by aiding the protein which is being synthesized to acquire a three-dimensional structure. So they act by refolding misfolded proteins or aiding in completely folded proteins. So they can reach their native conformation, go to the Golgi, and in the case of the membrane receptors, travel to the plasma membrane receptors to be able to be recognized by the agonist. So that agonist stimulation provokes internalization of the receptor. And then the receptor can be degraded or can recycle to the cell membrane. So that the thing is that it is very important to have these molecular chaperones that aid in that the receptor may take the third dimension, the three-dimensional configuration, and that they can travel to the cell surface membrane. But if the chaperones are not able to correct the misfolding, then they are targeted for degradation to proteasomes or to other structures. So misfolding may lead to degradation or intracellular accumulation with toxic effects, as I said previously. And there are some experimental techniques to aid these misfolded proteins to fold properly and to be targeted to the site of action. And there are physical methods. For instance, if we in the laboratory incubate at low temperatures, we may stabilize the conformation by preventing the association with some heat shock proteins, with some molecular chaperones. We may add also some chemical chaperones, but these chemical chaperones are very nonspecific, and they may rescue a lot of proteins, and many of which were whose fate was to be degraded. And we can do also genetic modifications. So if we do genetic modifications of a mutation, if we correct a mutation, then we can correct the folding of the protein, because in disease, the frequent causes of disease due to misfolding is because there is a mutation. And we finally may use pharmacological strategies, which we call pharmacoperones. So using the same natural example, the natural model of molecular chaperones, we can now use pharmacological chaperones to get the same action, stabilize the three-dimension of the protein, the three-dimensional configuration of the protein to be correctly folded and travel to the destination within the cell. And this is how the pharmacological chaperones act. Well, pharmacological chaperones are in general small molecules. They can be agonist, they can be antagonist, or they can be allosteric modulators. But the main feature is that they have to cross the plasma membrane. And second, they have to be highly specific for the target protein, so that they bind so that they may be able to bind exclusively the target protein. This is the pharmacological chaperone, binds the target protein, and then corrects folding and allows the protein to pass the quality control and to go to the Golgi and to be expressed in the membrane, where it interacts with the natural agonist. So that's the function of the pharmacological chaperone, small molecules that rescue function of a misfolded protein, in this case, of this membrane GPCR. So we have many kind of mutations, and these mutations may affect in different ways the function of the receptor. The type 1, the receptor is synthesized inefficiently because there are some mutations in critical regions of the gene that impedes the correct synthesis of the protein. And in type 2, Type 2 is the defective trafficking, where the protein is misfolded, and it cannot reach the final destination in the cell membrane. And then type 3 is when there is defective binding of the agonist, and then class 4 is when there is defective signaling. The receptor is fine, but there is a mutation which impacts on signaling. And any of these kind of mutations provoke what we call loss of function mutations and loss of function diseases. Okay, so the class 2, which are the trafficking defective mutations, they are very common. I mean, more common than previously suspected, because we didn't know exactly what misfolded means and how the quality control system of the cell work and how we're working the molecular chaperones and so forth. So it's quite frequently, and they are mainly due to point mutations in the gene that encodes for that protein. Now, if the mutations does not involve amino acids which compromise the binding of the agonist or activation of G proteins, then they can be corrected by pharmacological chaperones. So there are many diseases, endocrine diseases, I'm just talking about endocrine diseases, that are due to misfolding of the receptor, of the GPCR. Including nephrogenic diabetes insipidus, which is an X-linked disease, and in which the patient cannot manage properly water and electrolytes, and the patients die very early in life during childhood. Then, fertility problems, hypogonadotropin hypogonadism means that there are gonadotropins, low levels of gonadotropins because of absence of the gonadotropin-releasing hormone synthesized in the hypothalamus, or when there is absence of the gonadotropins because of defects in synthesis, etc., or of the receptor, of course, mutations in the receptors. Then hypothyroidism, congenital, and other alterations like hypocalciuric hypercalcemia, familial glucocorticoid resistance, and morbid obesity, and so forth. These are all caused by misfolding of the receptor. This is a representation of the gonadotropin-releasing hormone receptor. We have been working for many years with this receptor, and we use these mutations in which the glutamic acid is replaced by lysine in position 90 of the receptor. These all are mutations, loss of function mutations, and this was the first mutation in which we found that the problem is that the receptor is misfolded. It doesn't have a 3D configuration that allows to traffic properly. And we found that this mutation, in fact, provokes misfolding of the protein. This is the family that Dr. Mendez, we collaborated with him in Mexico, found a family with this mutation in which glutamic acid is replaced by lysine, and the patient has low gonadotropin-releasing hormone response, the hypothalamic decapeptide, low gonadotropins, and absence of puberty, and of course infertility. So this is the mutation. And these are other mutations that were reported in the literature, and they were tested in vitro in a cell culture of COS7 cells. So when the cells were stimulated with the agonist, buserelin, the agonist of the gene receptor, some of them did not respond to the stimulation by the agonist, including the NREK mutation, and others responded partially. This is the normal receptor, which is called the wild-type receptor. And what happened when we added the pharmacological chaperone, a small plasma cell membrane permeable small molecule, which is an indole, and we incubated before adding the analog, the agonist. And what happened is that almost all of the mutations tested recovered function in terms that they went to the membrane and they responded to agonists. Some of mutations were dead. They did not respond to any to the pharmacochaperone because the conformational defect is so severe that cannot be corrected. The protein cannot be stabilized by the chaperone. But the most important finding that the late Michael Cohn in 2013 published in PNAS was the proof of principle employing mice who had the mutation in the position 90 of the GNRS receptor. So he made a knockout mice and then treated with a chaperone, which was in-3. So the animals were kept in a cage where they can move freely. They have a catheter implanted subcutaneously, and then in-3 was administered subcutaneously every 6 hours at a constant dose for 30 days. After 30 days, the male mice were euthanized, and this is what they found. So these are the tests of the mutant animal. So small testes, atrophic seminiferous tubules where this spermatogenesis occur, very atrophic. This is the normal testes injected just with placebo. You can see here the accumulation of sperm, nest of interstitial cells, which are the responsible for testosterone synthesis. This is a heterozygous, which is completely normal also. This is the homozygous for the mutation, and this is the animal treated. So after 30 days, you can see that there is sperm formation, and the size of the tubules, the diameter is wider than the untreated animals. And biochemical studies show that this is the normal receptor in terms of the activity of an enzyme which is necessary for testosterone biosynthesis. And this is the heterozygous, this is the homozygous animal, and this is the animal treated with the pharmacological chaperone. And the same was observed with testosterone biosynthesis in these animals treated for 30 days. Then we analyzed if there is a possibility that shorter periods of treatment were sufficient, and just 8 days of treatment were sufficient to see in the testes of these animals some degree of spermatogenesis. So it was wonderful to find that this indole, this small molecule, may act in vivo. Of course, that was not the first in vivo experiment. There was studies in humans in which the group in Canada gave to children with diabetes insipidus a pharmacological chaperone and reestablished the water and electrolyte equilibrium, but unfortunately, due to toxic effects, that study has to be stopped. That was the Bernier in Canada. And another example is the follicle-stimulating hormone receptor, which is essential for follicular maturation in the ovary. So we have been working some mutations. Here are mutations. This part of the receptor is out of the cell. This is in the membrane. And this is inside the cell. And we have been working with a different pharmacological chaperone. And this mutation is the first mutation in this receptor, which was described in a large group of patients in Finland by our friend Ipo Utaniemi in 1995, very good friend of Manuela too. And this is a very nice control, and there's another mutation very close to the 189 mutation, which is the 191 mutation, who was described by a colleague of Manuela in Germany. And then we have another mutation here, but look at that this mutation is in the transmembrane region, here alpha helices, and these mutations are in the domains which are in the membranes, which is the 408 and the 419. All these mutations cause infertility, absence of puberty, absence of sexual development, and so forth. Okay, so this is how the group of Filpo found that the receptor is not expressed in the membrane. This is the normal receptor, and then when the antibody is permeabilized, I mean, the antibody against the receptor is allowed to cross the plasma membrane. In normal cells, the antibody is here, is retaining the cell surface membrane because the receptor is in the membrane, and in the patients affected with the mutation, the receptor is absent in the surface. We confirm these findings by another technique, which is flow cytometry, and see how the normal receptor is mainly extracellular, and a few amount of receptor is intracellular, and this is the mutation, is mainly intracellular, the receptor. And some years ago, we tested a allosteric modulator with Michael Cohn, and we found that the 189 mutation was rescued partially in the presence of this allosteric modulator, which was originally designed as an allosteric modulator for the luteinizing hormone, not for follicle stimulating hormone. And it was surprising that it could rescue this mutant receptor and also the wild-type receptor, which means that the normal receptor is normally synthesized inefficiently because it has some degree of misfolding. And then we recently, what did is to work with a small molecule manufactured by Canwell Pharmaceutics, and which is called 1405. And this is the receptor, the FSH receptor. The extracellular domain has been truncated, and we did some what we call the docking techniques and molecular modeling to see where this pharmacological chaperone binds, and we found that it binds to the external region. In the upper half of the receptor. It doesn't look so clear here because of the position of the receptor in this particular photograph, but it binds to several residues mainly in the transmembrane 2 and 7. And we have a technique to analyze if the receptor responded to the pharmacological chaperone. If the receptor, which is trapped inside the cell, respond to the pharmacological chaperone. And we use this Western blotting, which is a very frequently used biochemical technique in which the antigen, in this case the receptor, is separated by electrophoresis in polyacrylamide gels, then transferred to membranes, and then challenged with a specific antibody. And after addition of a second anti-IgG antibody, we may see where is the receptor by autoradiography. Okay, so this is the mature receptor, the FSH receptor. This band is the normal receptor, the wild-type receptor. This band here is the intracellular immature receptor. And you may see how the normal receptor, the wild-type receptor, increases when it is incubated with CAN 1405. And the mutation for O8 is also rescued by this pharmacological chaperone. Then we can measure the densities of these bloods and see how the mature receptor increase after incubation with the pharmacological chaperone. The 199 mutation does not respond because the chaperone is bound to this transmembrane region, changes the configuration, and does not affect apparently the extracellular domains where the mutation 189 is located. And also these receptors recover function. Look at this, which is partially active. It recovers function just as the wild type receptor. And the same findings were observed for the 191 mutation, which is very close to the 189 mutation. No rescue is observed. And the 419 mutation is rescued exactly as the same for 08 mutation because it's located in the same domain. So that this is interesting because it serves as a paradigm for other diseases that can be cured by treating with pharmacological chaperones. And there are pharmacological chaperones that are experimentally tested and for the angiotensin 1A receptor, for the melanocortin-4 receptor, whose mutations cause morbid obesity, for the calcium sensing receptor, for the vasopressin receptor, for the prokineticin receptor, generase receptor, which provokes hypogonadism, as well as for the follicle-stimulating hormone and the luteinizing hormone receptors. So, these are just chaperones that have been tested successfully for some mutations due to misfolding of the corresponding receptor. So, in conclusion, chaperones of the quality control system of the cell play a critical role in keeping protein homeostasis, proteostasis. Misfolding of proteins as a result of mutations lead to conformational diseases, which may be these diseases that I mentioned here, that I described here, endocrine diseases, but can be neurodegenerative diseases, which are more important: Alzheimer, Parkinson, diabetes mellitus, and other diseases, Huntington disease, and so. Misrouted receptors causing disease can be rescued with small pharmacological chaperones and restore function, at least experimentally. And not so experimentally, because cystic fibrosis, which is a not so infrequent disease, there is a mutation that is prevalent in the United States, in the population in the United States, which is due to a deletion in the amino acid 509. And there are reports that by the use, and the problem is that the channel, the chloride channel, is misfolded. It cannot go to the surface, to the membrane surface. So there's a chaperone or class of chaperones that take the cystic fibrosis channel and regularly and take it to the membrane. And then a second chaperone is added to prevent degradation after internalization of the channel. So this is a reality. Cystic fibrosis, at least the 509 mutation, can be corrected by the administration of chaperones, and the kids with this mutation improve the quality of life, improve several clinical parameters, respiratory parameters, and so forth. So chaperones for many diseases are the solution to this kind of conformational diseases. And now, after the talk that we listened today about the molecular machines, then it's conceivable that these small molecules can be administered orally be transported to the target cell specifically and cross the membrane. They have to be permeable and correct the misfolded protein to allow it to traffic to the site of action. So high-throughput screens are used now to identify thousands of chaperones, and it's a valuable rapid testing massive numbers of compounds that existed many years ago, but that they were kept in files of, in banks of molecules that didn't work because they were permeable. So the objective of the antagonists and agonists were that they shouldn't cross the membrane, but now we found that if they cross the membrane, they can rescue the receptor, which is intracellularly trapped. So they can rescue and aid in a number of diseases caused by GPCRs. Now, I just would like to thank the student officials for the organization of this fabulous transdisciplinary meeting, particularly to Aurélien Montagu, who responded all my questions and my concerns. Thank you very much, Aurélien. I would like to also a special thank to Professor Paul Vignier. Professor Paul Vignier accepted me as a scientist, as a business scientist in 2009 and 2010 at the laboratory of Eric Reiter in the era in Nusigi. And Eric and I thought that it could be very interesting to create consortia with investigators, with scientists from outside and inside Europe. So Professor Vignier accepted very enthusiastically this idea, and the first consortium was created. We called it initially ESCARTS. Then the names changed and so forth. But it was a student consortium. And it was conformed by Eric, myself, Il Poutaniemi from England, Jim Diaz from Albany, New York, and George Busfield from University of Wichita in Kansas. And we came here every year to discuss experiments, to show results, and to write papers. And this consortium culminated with the organization of the third International Congress in gonadotropin and gonadotropin receptors, and 3 years later, Manuela organized a fantastic meeting in Modena, Italy, and then we have not been able to organize another international congress because of the damn COVID-19. I would like also to thank my collaborators in Mexico. Eduardo Jardón is the one who does the molecular dynamic simulations, and these are my technicians, molecular biologists, Teresa Sariñán and Ruben. And I will show you just in the next minute my wonderful university. This is the library of the university, and the four walls have micro mosaics in the form of a mural describing the development of science. This is a beautiful mural built by O'Gorman, a Mexican muralist. And this university has a wonderful cultural section with a contemporary museum, which is this, Museum of Art, and a very nice auditorium which hosts the Philharmonic Orchestra of the National University. And thank you very much for your attention.